Welcome to the summer feedback for A-Level Biology, Specification B. This pre-recorded event for Pearson Biology Specification B is for teachers and looks at how students performed in the summer 2023 exams. It will provide guidance on how to address feedback in the examiner's reports. You will receive feedback on the national performance of candidates in A-Level papers 1, 2 and 3. Look at the examiner's report and address common issues and facts. I will also cover areas of overall strength and weakness, both in subject content and in skills, for example maths and practical work. We will first look at the overall review of performance of candidates across all three papers. We will follow that by reviews of candidate performances on papers 1, 2 and 3. And finally, look at a summary of the key themes that have emerged. First, we're going to look at an overall review of candidate performance across all three papers. This year, grading returned to the standards of 2019. As time has gone by, senior examiners have noted that the quality of response from candidates that are representative of A star to B grades has improved. This is an expected and welcome trend, and is due to centres and candidates becoming more familiar with the styles of question and the demands of the specification. The quality of responses from candidates representative of D and E grades has improved, but not as much as the A star to B grade. In 2023, the overall standard of candidate response was very good and showed an improvement on previous years. Candidates and centres should be commended on all their hard work, which has often been in difficult circumstances due to the government responses to the coronavirus. This cohort did very well, especially with most not sitting external exams for GCSE. So many candidates would not have experienced carrying out exams under pressure. Candidates representative of A star to B grade showed a further improvement on 2019 with the quality of their answers. Candidates representative of D to E grades, however, still find the papers challenging. We will now look at some of the common errors made by candidates across all three papers. Some candidates did not read all the details given in questions. Most of the information in a question is there for a reason, and candidates should do their best to try and read and digest all the information given in the question. Some answers lack detail, and candidates sometimes use words and terms imprecisely or inaccurately. Candidates should always be careful to give as much detail as possible and use scientific vocabulary accurately and precisely. Not including information from all data when answering longer, level-based questions was also a common error. Candidates should explore data fully when being asked with command words such as discuss or evaluate. Some candidates misinterpreted command words. A common mistake is to simply describe data when an explanation is needed. Candidates should be very careful to make sure that they are familiar with all the command words listed in the specification. Some candidates left questions blank. Candidates should always be encouraged to attempt every question. With calculations, they should make some attempt at them. There may be marks awarded for working, even if the final answer is incorrect. Some candidates did not complete the papers. Candidates should make sure that they do not spend too long on any one question. They should also always focus on the number of marks in a question rather than the number of lines. Sometimes there are many lines for a question. Some candidates have large writing, and the extra lines are often there to take account of this. It is very important that candidates under the styles of questions used in examination papers. All questions have a command word that has specific meanings. Candidates should be familiar with all the command words that are listed in the specification. In 2023, it was noted that candidates are becoming more familiar with the design of the papers and the requirements of the command words. Some candidates, however, find certain command words challenging. Compare and contrast, for example, requires candidates to give both similarities and differences. Some candidates did not do this and only focused on one aspect. Analyze the data is the start of many questions. It is not a command word, however, but an instruction to use data in answers. The command word often comes later, such as analyze the data to explain something. The command word evaluate requires candidates to invest both sides of an argument, or to consider evidence that both supports and does not support a conclusion. The command words comment on is often found difficult. This requires a full description of all data patterns in a question and some evidence of a judgment. Many candidates often focus purely on the factual content of the specification. They should also appreciate, however, that there are 3 assessment objectives, which have different weightings on all 3 papers. These 3 assessment objectives are AO1, which is where candidates need to demonstrate a knowledge and show understanding of scientific ideas, processes, techniques and procedures. This is like factual recall and is often liked and looked forward to by candidates who feel they have revised thoroughly and prepared well. AO2 is where candidates apply knowledge and understanding of scientific ideas, processes, techniques and procedures. This is all about applying their knowledge to data information. Questions can use unfamiliar data, can ask for calculations or graph drawing. AO3 is where candidates analyse, interpret and evaluate scientific information, ideas and evidence, including in relation to issues. This is all about making judgment and the assessment of practical skills and understanding. In 2023, many candidates prepared well and showed excellent factual knowledge across all 3 papers. Some candidates were less confident when handling AO2 questions and when analysing unfamiliar scenarios. And sometimes candidates feel to lack confidence when handling calculations. Many candidates showed excellent understanding on practical methods for AO3 on paper 3, but there were a few who did seem to have limited practical experiences. The weighting on the assessment objectives across the 3 papers is slightly different. We should note the difference that AO3 has a higher weighting on paper 3, and it is only on paper 3 where practical methods are assessed. We will now look specifically at paper 1. Paper 1 consisted of a range of question styles, including multiple choice questions, shorter 1, 2 and 3 mark answers questions, some longer 4 mark questions, and as always 2 6-mark level-based questions. In general, the multiple choice questions were quite well answered, and calculations were performed much better than in previous years. It was also evident that centres are more familiar with the requirements of the level-based questions and are preparing their candidates for these far more effectively. Now we will look at some specific examples of candidate answers for paper 1. This question here, question 1b, is an AO2 question. Candidates have been presented with the results of an experiment on potassium ion uptake by plant seedlings in anaerobic and aerobic conditions. The graph clearly shows a fall of potassium ions in anaerobic conditions and an increase in aerobic conditions. Information is also given inside the question. And the first bit of the question here is very important for candidates to understand in their answer. Candidates, when presented with data such as this, should think about what a question is testing. It is clear that the question is about membrane transport. It is also clear that aerobic and anaerobic respiration are being tested. Many candidates found this question quite challenging. Many simply described the data and did not explain it. When we look at the question, we can see the question is clearly explain the changes in the concentration of potassium ions in the cells in the roots of the seedlings during an investigation. And many simply gave a description of the graph going down and then going up again. Many candidates did also not refer to the roles of oxygen in aerobic respiration, or recognise that active transport must occur as the uptake increases when there is oxygen present. Few referred to diffusion of ions out of the cell over the first part of the graph. This is the mark scheme for the question. What we should do is note the accurate terminology that is listed inside it, such as active transport for mark point 3 and diffusion for mark point 2. If we look at the mark scheme, we can see what it's looking for. Mark point 1 is looking for an idea that potassium ions leave the cell or are used, and in addition that there is an increase in concentration due to potassium ions being taken up by the cell. This is not simply a description of the data. Mark point 2 asks for an explanation that facilitated diffusion is occurring, and that also active transport must have occurred. Mark points 4 and 5 focus on the idea that when there's oxygen present, aerobic respiration occurs, and then finally that aerobic respiration will generate more ATP, which should cue candidates into the idea that active transport is occurring. This candidate answer is an excellent answer that gained 4 marks. The candidate begins their answer at the top with a description of what's happening. This is good practice really, and often candidates should be encouraged to describe the data. There are no marks available for this. However, it does then set the scene for leading them on to an explanation. In this question, the candidate clearly states that potassium ions leave the cell by facilitated diffusion, that ion uptake is by active transport, that aerobic respiration released the most energy, and MP1, the first mark point about potassium ions leaving the cell and then being taken in, is clearly given throughout. The answer is detailed and accurate, and the candidate clearly shows accuracy in their writing by referring to potassium ions rather than just purely potassium, which is another common error seen by many candidates. This answer is a weaker answer that gained no credit. There is no actual explanation about the method of transport, which should talk about active transport or facilitated diffusion. There is a description of data, and we can see at the start the candidate talks about a decrease in concentration of potassium ions where they're taken in, but there's no real reference to them leaving or entering. There is a reference to oxygen later in the question, but the context is unfortunately incorrect. The candidate has mentioned aerobic conditions, but they haven't really explained that aerobic respiration occurs when oxygen is present. Question 3C.E3 is an AO1 question that asks candidates to describe the method by which sugars are transported. If we look at the question, it clearly says, describe how sugars are transported through the phloem. Many candidates found this question challenging and did not include detailed descriptions referring to sources, sinks, or hydrostatic pressure. The question is an AO1 question and looks for a detailed description. It's a good example of how we need to use accurate and high-level language, and this will gain full credit. We should note how the mark scheme lists key terms that are needed. So mark point 1, we see a clear reference to mass flow is needed, or mass transport. Mark point 2, we have clear references to sucrose or sugar being actively transported into phloem at the source. Mark point 3, we're looking for an idea that sugars or sucrose move out or diffuse out of the phloem into sink tissues or storage sites. Mark point 4, we're looking for a description of the entry of water at the phloem, into the phloem at the source, and then its loss at the sink. And the final mark point, we're looking for the idea of the sugar moving from a region of lower hydrostatic, note that hydrostatic is underlined, pressure, or candidates would have stated moving from a high to a low hydrostatic pressure. This is an example of a very good answer that gained 2 marks. The candidate clearly states that mass flow moves the sugars in line 1. The candidate also describes the movement of water in and out of the flow. Pressure is mentioned, however it is not hydrostatic pressure, and hydrostatic was a word that was underlined in the mark scheme, and there is no reference of it moving from a higher pressure or moving to a lower pressure. Question 6b. This question is also an AO1 question that asks for an explanation of the importance of thylakoid membranes in the light-dependent stage of photosynthesis. Strong answers used key vocabulary and addressed the command word in the question, which was explain. Weaker answers simply described the light-dependent stage and did not relate this to the thylakoid membranes. The mark scheme again requires accurate vocabulary. If we look at the mark points, we will notice that many of them have the words "so that" inside those mark points. The words "so that" show that the question requires an explanation. Mark point 1, we are looking for the idea that photosystems, or PS1, PS2 or chlorophyll, or a named pigment, are present in the membranes so that light or photons can be absorbed. Notice the word absorbed is underlined. Mark point 2 requires an idea about electron carrier proteins or the electron transport chain is present so that reduced NADPH is formed, or hydrogen is pumped into the thylakoid space. Mark point 3 requires an idea of ATP synthase or ATPase being present so that hydrogen ions can pass through. Mark point 4 requires an idea that the membranes enclose a space or form a barrier so that hydrogen ions can accumulate. The final mark point is an idea that is given for the idea that membranes are a site of photolysis or chemiosmosis. Note that most of the mark points, however, have the words "so that," and this is because this question is an explain rather than a describe their importance. This answer is a very strong answer that gains 3 marks out of the 4. The candidate clearly gives an explanation throughout in their answer rather than simply a description. Mark point 2, 3 and 5 are all evident in this answer. Mark point 1 was not awarded. This mark is about the pigments or the photosystems absorbing light. The word absorbed was underlined in the question, in the mark point. And in this answer, there's no mention of absorption of the light. Candidates should be very, very careful when writing their answers to use specific, accurate and precise language. Many candidates refer to pigments attracting light, which is not quite right. We were looking for the idea of absorbing light. Many candidates also mentioned the electron transport chain, but gave very confused answers in how it leads to ATP production rather than giving keywords in the correct context. It is not enough just to put the keywords in, they need to be there in an accurate context. Question 7b, part 4. This is a 6-mark level-based question. The question is all about speciation of different species of fish. Information about the 2 species of fish was given to candidates at the start of the question. And if we look at this part of the question, we can see it clearly asks how 2 species of fish evolved to become separate species. Many strong answers were seen that fully explained allopatric speciation. A few candidates wrote about sympatric speciation. In this question, information was given to candidates all about the separation of the species in separate ocean trenches that were long distances apart. Strongest answers that gained 5 or 6 marks related the speciation to the context of the question. Some candidates gave answers that just simply talked about speciation in generic terms. Candidates should always be reminded to apply their knowledge to the actual context of the question. Weaker answers tended to confuse both allopatric and sympatric speciation. Many gave insufficient depth, and they often, as already mentioned, did not relate the speciation to the context of the question. This is the mark scheme for the question. There are 5 aspects that refer to different ideas. In level-based questions, indicative content is listed in the mark scheme. This is not exhaustive, and candidates may mention other relevant facts. It's also true that candidates do not have to mention every mark point that's listed in the indicative content. In this mark scheme, there are 5 aspects. Aspect 1 is about the isolation of the species of fish in the 2 areas. This is about allopatric speciation. Aspect 2 is about the selection pressures, and this is about how the 2 areas were different. Aspect 3 is this natural selection story. What we're looking for here is a detailed description and explanation of how natural selection would occur in the 2 areas. Aspect 4 is about a decrease in gene flow and reproductive isolation. And aspect 5 is about epigenetics and how in some areas epigenetic changes may have occurred to alter gene expression. The levels were awarded. Level 1 was awarded for 1 mark for any relevant comment, 2 marks for a demonstration of a proper understanding of one aspect. For 3 marks in level 2, candidates needed to show a good understanding of one aspect and a mention of one of the other aspects. For 4 marks, a good understanding of 2 aspects was required and 1 other aspect mentioned. For 5 marks, candidates needed to show a good understanding of 2 aspects and 2 others mentioned. And for 6 marks, 3 aspects needed to demonstrate good understanding and another aspect mentioned. And there also needed to be detail relating to the scenario of the question, rather than just a generic context. Most candidates were able to mention aspect 1 and aspect 3. So most were able to talk about the isolation and the natural selection. There were often mentions of aspect 2 and aspect 4, but these were often with less detail. Very few candidates referred to aspect 5 about epigenetics. This is a candidate answer to question 7b. This is a very strong answer that gained 5 marks. The candidate clearly shows a very good understanding of 2 aspects, which are the isolation, all about the allopatric speciation. We can see that in line 2, and natural selection. There is also detail about selection pressures and gene flow. With level-based questions, candidates should explore topics as fully as possible in order to gain maximal credit. Question 8b. This is a compare and contrast question. Candidates were asked to compare and contrast the humoral immune response with the cell-mediated immune response. To gain full marks, there need to be both similarities and differences in all compare and contrast questions. For this question, strong answers tended to have separate sections clearly stating similarities and differences. Candidates should be encouraged to have separate sections on all compare and contrast questions that show all the similarities and then all the differences. Weaker answers tended to describe each response without stating clearly the similarities and differences. So a weaker answer would often describe the humoral response and then describe the cell-mediated response. The question was looking for candidates to point out clear similarities and differences. Another common error was to refer to the role of T cells rather than specifically referring to T killer cells and T helper cells. If we look at the mark scheme, it clearly shows how it's arranged to reflect the idea that there's a similarities section and a differences section. For similarities, we see marks are awarded for recognizing that both are forms of active immunity, both involve T helper cells, both involve cytokines, and both result in the production of memory cells. For the differences, what the examiners were looking for was an idea that antigen presentation in the humoral responses by B cells and in the cell-mediated response, infected cells present the antigen. Now notice again, for differences, we need to have both sides of the story. There's another mark for stating that B cells are involved in the humoral response, but not in the cell-mediated response. Again, a candidate can't just say that B cells are involved in the humoral response without a reference to the cell-mediated. An alternative to this was stating that T killer cells are involved in the cell-mediated response, but not the humoral response. A mark was awarded for stating that antibodies were involved in the humoral response, but not in the cell-mediated response. And there's a mark for stating that in the humoral response, antigens or pathogens and infected host cells are involved, but in the cell-mediated response, it's host-infected cells. Again, it's very clear from the mark scheme for differences that candidates should always give both sides of the argument. This is an example of an excellent response to question 8b. 3 marks are awarded for this. It's very clear that the candidate has stated that both involve T helper cells. It's very clear that the candidate states that only the humoral response uses B cells and that the T killer cells are used in only the cell-mediated response. The candidate clearly points out at the bottom here of the answer that both produce memory cells. A mark was not given for antibodies being affected in the humoral response, as no statement is given about their lack of involvement in the cell-mediated response. This is a common error made by candidates when pointing out differences, to only put one side of the argument. Both sides need to be mentioned to gain credit. This is an example of a much weaker response to question 8b. This answer did not gain any credit. When we look at the answer, we can see the candidate has written some detail, such as cell-mediated response causes T killer cells, but there is no actual comparison and no contrasting in the answer at all. It is not enough in compare and contrast questions to simply give descriptions. There needs to be direct comparison of both sides. Candidates should be encouraged to do a section at the start on similarities and a section on clear differences, and in the differences refer to both sides. For example, in generic terms, X has something, but Y does not have something. We will now look at some of the responses for paper 2. Paper 2, like paper 1, also consisted of a range of styles of questions, including multiple choice questions, shorter 1, 2 and 3 mark questions, some longer 4 mark questions, and again 2 6-marked level-based questions. Most questions were well answered. Most candidates demonstrated strong knowledge of all topics covered. Some candidates, however, gave basic descriptions for level-based questions without fully exploring all the information given. Candidates should be guided by mark allocation rather than numbers of lines. Many answer lines are often given for questions, but they do not necessarily need to be filled. Writing too much can take up valuable time in examinations. Now we will look at some specific examples of candidate answers to different questions. This is question 2C.E2. The context of the question is about an enzyme called bromelain and how copper sulfate affects its activity. It's an AO2 question and candidates were required to apply their knowledge to data about the enzyme activity. Bromelain is probably an unfamiliar context for most candidates, but the data is given, so candidates should be able to apply their general knowledge of enzyme to this question. Generally, strong answers recognise that copper is an inhibitor. Candidates were here asked to explain the effect of copper sulfate on the rate of digestion and they were given a graph which showed the different levels of copper sulfate included. The very strongest answers recognised it was non-competitive and went on to explain the results rather than simply describing them. Weaker answers often suggested that copper was a competitive inhibitor. Competitive inhibitors eventually their effect would lessen as the concentration of soluble protein would have increased. Some candidates simply gave descriptions, and again, it's very important for candidates always to read the command word. This question asks for an explanation, not a description. This answer gained 1 mark for recognising that less binding of enzymes and substrates. Notice how the candidate says this later on in the question, on the last line, that they recognise that less enzyme substrate complexes will form. Unfortunately, the candidate incorrectly states that the inhibitor is competitive. This was a very common error. Candidates should be familiar with inhibition both as competitive and non-competitive and be able to recognise the effects of both types of inhibition. Candidates are encouraged to use terminology such as enzyme substrate complexes. This answer shows this at the end, and this is good practice. Many candidates gave vague terms about enzymes colliding with substrates. It's good if they can use the key terminology. This is question 2b. Now, this is a calculation question. And in A-level biology, 10% of marks are required to be from what are called level 2 maths questions. Level 2 maths questions are higher standard GCSE maths questions. This is an example of a 3-mark question, and it required candidates to calculate the volume of an area of agar that had been digested by an enzyme. Because this is a 3-mark question, there will be several steps involved in the question. In this one, these steps included converting units, calculating the volume of 2 cylinders, the initial volume of the disc that was cut out, and the final volume where the agar had been digested, and then subtracting the 2 volumes. The answer was also asked to be given to 2 significant figures, something that many candidates missed. Candidates should always show working. Credit may be obtained even if the final answer is incorrect. Common errors in this were only in calculating the volume of the final cylinder. Many candidates forgot to subtract the initial volume. Many candidates didn't give the answer to 2 significant figures. And some did not convert between units correctly. Incorrect conversions between units is often a very common error across all papers. This is an example of an answer that gained 1 mark for correctly calculating the volume of one of the cylinders. The candidates forgot here to calculate the initial volume and subtract it. 28,000 was actually a common answer and showed that many candidates did actually forget that this initial cylinder had been cut out of the agar. This is an example of an answer that gained all 3 marks. What is also good about this is all the working is clearly shown. This is good practice. Candidates should be encouraged to show all the steps in their calculations. If their final answer is incorrect, they may still gain credit for the working. We'll now look at question 4b.E2. This question is all about salt marshes, succession, and how changes occur in a salt marsh with the age of the salt marsh. The question is actually an AO2 question because it requires candidates to look at unfamiliar data about species distribution. Information was given in the question to cue candidates into the idea of succession. Candidates were shown, and we can see this on the second line, as the distance from the sea increases, the age of the salt marsh increases. This was to try to cue candidates into the idea that succession is part of this question. The final question is, explain the changes in distribution of species as distance from the sea increases. Strong answers tended to correctly explain how succession would have created the results and always related the process to the context of the salt marsh rather than just giving a generic description of succession. Weaker answers often just described the data or gave generic descriptions of succession and did not relate it to the data given to them. This candidate answer is a very strong answer that gained 3 marks. If we look at the answer, it clearly refers to the process of succession in line 4 down here, and clearly goes on to explain about samphire being a pioneer species and colonizing the area. The candidate later goes on to explain that abiotic conditions change and affect the distribution of species. We'll now look at question 7b. This is a level-based question, and it presented candidates with data about the effects of seed banks on genetic diversity of plant species. 2 graphs were presented to candidates. One shows genetic diversity of plants in the wild, and then seed banks. The second graph over here shows differences in DNA sequences of samples of seeds in seed banks compared with wild plants and how different they are, and looks at them at how long seeds have been in a seed bank. The command word of the question is actually discuss. Candidates are first asked to analyse the data. Analyse the data is a cue that candidates have to make to use data in their answers and really come to some judgment. In this question, they have to discuss the results. Notice that error bars are given on the graph. And if we notice on this graph, the line of best fit through them often has data points that are quite far from the line. Now, if there is a hint to candidates about data quality with error bars, overlapping error bars, and data points that may be away from lines of best fit, it is a cue to them to show that we are looking for some idea of making a judgment based on quality of data. If error bars are on a graph, candidates should really try to refer to overlaps and what this means, or not having overlaps and what it means. If we have a line of best fit and there are many points away from it, or they're clustered, again, candidates should make reference to the quality of data and how well they can make a judgment from it. Strong answers that gained 5 or 6 marks included descriptions of data, explanations, and comments. And we see this here in the way that levels were awarded. Level 3 required a discussion that had some degree of description of the data, a degree of explanation, and something about the comments about validity or quality of data. To get 6 marks, both graphs had to be referred to. Weaker answers tended to merely give a description, and so were restricted to level 1. There were many excellent answers, and examiners were generally very impressed with how well that centres are clearly preparing candidates for longer data analysis questions. And many candidates did appreciate that they needed to look at both sets of data and look for descriptions, explanations, and comments about validity. This long-looking answer here gained 4 marks. It is a good solid answer, and the candidate did explore the information well. There are lots of descriptions about the data that we can see woven throughout, and they describe both sets of data. The candidate has also gone on to give some explanations for the reasons for the differences in variation. However, there are no comments about validity or data quality or overlapping error bars or the line of best fit. Because there's no mention of validity, the candidate is restricted to level 2 and so gained 4 marks. This is another answer that only gained 2 marks. The candidate has attempted the question, but only gives descriptions throughout of the data. In a discussion question, we expect candidates to explore questions and data fully. There is descriptive work in here, but there is no explanation and there is no comment on validity. So this answer gained 2 marks. We'll now look at question 5C.E1. Classical genetics in topic 8 is a feature of paper 2. This question is a typical genetics question that uses a pedigree diagram. This question is actually quite a challenging question and required candidates to deduce a mechanism of inheritance of a form of colour blindness from this pedigree diagram here. Notice deduce, which is an unusual command word not often seen. Deduce requires candidates to work something out and always show their working. To help them with this, included in the question is a statement for them to use the information to support their answer. Many excellent answers were seen to this that clearly stated that the allele is recessive and not sex-linked, and went on to give it a reason for both. Weaker answers tended to guess that the colour blindness was sex-linked and often gave no cross that showed this. This is a candidate answer for 5C.E1. This is an excellent answer that gained all 3 marks. We can clearly see that the candidate states that the allele is recessive on line 2, and they then go on to link this to a cross which demonstrates this. At the end of the answer, the candidate states that it is not a sex-linked condition. They haven't given a reason for this, but they've already got 3 marks. Most candidates were able to recognise that the condition is recessive. However, fewer went on to state that it is not a sex-linked condition or is autosomal. We'll now look at paper 3. Paper 3 is slightly different to paper 1 and 2. It's a longer paper and it consisted of a range of question styles, including shorter 1, 2 and 3 mark answer questions, longer 4 and 5 mark questions, and 1 9 mark level based question. Notice there are no multiple choice questions on paper 3. Paper 3 always has a slightly different emphasis compared with papers 1 and 2. And we will notice if you look at the start, the balance of assessment objectives is slightly different. Paper 3 has contents from all 10 topics, unlike paper 1 and 2, which are restricted to topics 1, 2, 3 and 4 on both, but paper 1 having topics 5, 6 and 7, and paper 2 having 8, 9 and 10. Paper 3 has information from all 10. Paper 3 also assesses practical skills and understanding that are not assessed on paper 1 and 2, and that accounts for the higher level of AO3 on paper 3. General comments for paper 3. Questions were set on 6 of the core practicals. Many candidates were able to recall the methods followed as well as being able to explain the reasons behind them. Several indirect practical skills were tested. Now these include things like commenting on experimental design or evaluating methods, writing a null hypothesis, identifying variables to be controlled, plotting graphs, processing and analysing data, and applying their scientific knowledge to practical context. These were often answered well by many candidates. There was a more mixed response to questions with math skills on this paper. Candidates would generally benefit from more practice in several areas, including making up dilutions of stock solutions, how to calculate the biodiversity index, how to carry out and analyse results of statistical tests, and how to use the Hardy-Weinberg equation. And it was clear that some of the advice given in the 2022 examiners report has been implemented by centres, and centres should be commended on the way in which they have prepared their candidates for this exam. The practical skills questions that were found most challenging were 4A.E1, which was a question about how to make a dilution of gibberellin. 4B.E2, a question which asked for a description of 3 ways of improving a practical. And question 10B, a question requiring a justification, which is something candidates often struggle with, of 2 improvements to an experiment. In question 4A.E1, which is shown here, candidates were asked how to dilute a stock solution. If we look at that here at the bottom, we can see .E1, a stock solution of gibberellin has a concentration of 1 gram per decimeter cubed. The question then asks to describe how you would dilute this stock solution to give a test solution of 300 micrograms per decimeter cubed. Very few candidates gave correct methods, and we will look at an example of this later. Question 4B.E2 asks for 3 further improvements to a method outlined in parts A and B. This is again about the question on gibberellic acid. Unfortunately, few were able to give genuine improvements to the experiment that would actually improve validity. Notice that question wants further improvements, not things that were already in the method, and there was an emphasis on how this would improve validity. So generic improvements that would not improve validity were not accepted. Many candidates suggested an improvement, but not one that would improve validity. For question 10B, candidates were asked to justify 2 improvements that would ensure validity of an investigation into respiration by maggots. Many candidates gave descriptions or suggestions for improvements. However, very few recognized the command word justify. Justify requires a reason for those improvements. For example, we will always use the same species of maggot as species could affect the rate of respiration. It is very important to give a reason when a justification is asked for. We'll now look at some specific examples of candidate answers. We'll now look at question 1B. This question introduced candidates to polysaccharide, starch, and cellulose. Candidates then had to give 3 differences between the structures of starch and cellulose. Many candidates gained at least 2 marks. Weaker answers, however, often only describe the structure of one substance. For example, many stated that cellulose has beta-glucose, but did not go on to state that starch has alpha-glucose. Both structures should always be referred to. For example, starch has something, however cellulose has something different. Some candidates did not refer to structures and actually gave properties, such as stating that both are insoluble, or starch can be used for energy. The key in this question was differences and also structure. If we look at the mark scheme for this question, we can clearly see that all the mark points require a comment about both structures. For example, mark point 1 states that cellulose is made of beta-glucose, but starch is made of alpha-glucose. In mark point 2, we can see that cellulose contains unbranched or straight or linear chains, but starch contains branched chains. Mark point 3 states that cellulose contains 1,4 glycosidic bonds, but starch contains 1,4 and 1,6 glycosidic bonds. And in the final mark point, we see that in cellulose, the glucose inverts or flips every other molecule, but in starch, they're in the same orientation. It's very important to always give both sides of the argument. This is an example of an answer to 1B. This gains all 3 marks. 3 clear differences are seen. Alpha and beta glucose in the first line. The candidate also goes on to point the difference of branched versus straight chains, and then at the end explain that cellulose has 1,4 links, but starch has the 1,4 and 1,6 links. This is a good example of good practice where a candidate has given both sides of the story for each mark point. This answer gains 2 marks. There are correct descriptions of the glycosidic bonds at the start of the answer, where it says starch has 1,4 and 1,6 glycosidic bonds, but cellulose only has 1,4. And the candidate states that cellulose has beta glucose but starch has alpha glucose. No other mark was seen in the question though. We'll now look at question 4A1. This question tested both practical skills and math skills. The question, as we've already mentioned, was found to be very challenging by many candidates. Candidates were asked how to describe how you dilute a stock solution to give a test solution of 300 micrograms per decimeter cubed. The most common errors were candidates suggesting to add masses of gibberellin to volumes of water instead of explaining how they would dilute a stock solution. They needed to state, if we look at the mark scheme, that they would dilute the stock by a factor of 1 in 1,000 or a ratio of 1 to 999, and then some idea of a ratio of 3 in 10 or 3 to 7. Some candidates gained the mark point 2, but then didn't actually go on to mention to notice that they needed to do the 1 in 1,000 dilution. Many candidates did not seem to know how to perform dilutions. This is an indirect test of a practical skill, and candidates should be familiar with many practical skills. This is an example of 4A1, an answer to 4A1, and we can see this is a very good answer that gained both marks. The 3 in 10 dilution is given clearly at the start, where we can see they've got 3 and 7. And they've also gone on to explain how they would do the 1 in 1,000 dilution. This is an example of an answer that gained 1 mark. We can see that the candidate has correctly referred to the 7 to 3 dilution, but they have not gone on to explain how they do the 1 in 1,000. This was a common error. We'll now look at question 4A.E2. This question assessed candidates' understanding of Core Practical 14. Candidates should make sure they're familiar with all of the core practicals. Core Practical 14 is about assessing the effect of gibberellin concentration on amylase activity of seeds. There are also some general skills tested within question 4. On the whole, understanding of core practicals was generally thought to be very good by most candidates. There were, however, a minority of candidates who seem to have had limited experience of these core practicals. Centres should try to make sure that candidates have seen all the core practicals within the 2 years of the course. In this question, candidates were asked to read a method about Core Practical 14 and then state reasons for 2 of the steps in it. In this case, why the cereal grains were cut in half and half with the embryo discarded, and why the seeds were then placed into sodium hypochlorite solution for 5 minutes. This is the mark scheme for 4A.E2. What we can see in it for step 2, what the examiners were looking for was that candidates recognise that the embryo produces or releases or has the gibberellin, or an idea that the endosperm is the part that responds and produces the amylase. For step 3, we are assessing that candidates understand what the bleach or sodium hypochlorite does, and we're looking for an idea that it kills bacteria or sterilises the grain, or any idea that if there were bacteria they would produce amylase. For this question, most candidates gained at least 1 mark, often for stating that sodium hypochlorite sterilises the grain. Few, however, recognise that the embryo needs to be discarded as it's the part that produces a gibberellin. In the core practical guides, this is a key part of the experiment, that candidates have to cut the seeds in half and only keep half of them. This is an example of a typical answer that gained 1 mark. No mark was awarded for the first part. For step 2, the candidate says endosperm contains the starch reserves and there's no idea there about gibberellin from the embryo or the idea of amylase production by this. They have, however, got a mark for the idea that the seed was sterilised by placing them in sodium hypochlorite. We'll now look at another part of question 4. Question 4b.e3. This question tests candidates' understanding of how to modify a practical design to improve validity. Note further improvements are required, so restating ones already given in the question gained no credit. The question states, describe 3 further improvements to the method outlined in parts A and B that would improve validity of the results. Modification of practicals in general is a key feature of paper 3. We're also in this one looking for validity. So if we look at possible answers, we have the idea that the grain should be rinsed in water after soaking in hypochlorite. The idea that more than 1 grain for each concentration or repeats should be done. The idea of using aseptic technique so that there's no contamination. The measuring of a diameter several times. This is slightly different to the idea of repeating with more grains. Or the idea of a control with 0% gibberellin to see what would happen with no gibberellin. All of these mark points are further improvements, and they are all about validity. This is an example of an answer. This answer gained all 3 marks for giving 3 correct improvements that are all about validity. We can clearly see mark point 1. The candidates have stated that they would soak the grain in distilled water rather than gibberellin to act as a control experiment. They then gone on to say they would use enough grains to repeat the experiments 3 times for each gibberellin solution. So the idea of repeats, they've then gone on to say that they would calculate means as well. They then go on to say they would wash the grains with distilled water to remove sodium hypochlorite. This is a well-presented answer that clearly shows the 3 improvements that would improve validity. This is another example of an answer for 4b.e3. Now here we can see the candidate gains just 1 mark for the idea of repeats. And that's given here in the 3rd bullet point, the idea of repeating the experiment. The first mark point and the second mark point are not really relevant because they would not improve the experiment and they certainly wouldn't improve validity. We'll now look at question 5b.e1. Now 5b.e1, if we look at the question here, states candidates need to describe the method used to isolate individual species from a mixed culture of bacteria in nutrient broth. Now this question assesses candidates' understanding of core practical 13, a microbiology practical. The question actually asks for a very straightforward description. There's no justifications required or no explanations required. It's asking for a description how to isolate individual species, and there's a clue in the question that individual is required from a mixed culture. The quality of answers was rather variable. There were many excellent answers that showed candidates were very familiar with this practical. Others, however, did lack detail and did not go beyond setting up of the initial streak plate. Many did not go on to explain how they could isolate the individual species and only focused on half the question, the idea how you would plate bacteria out. If we look at the mark scheme, we can see it also requires suggested temperature and times for incubation, which is quite a bit of detail. The description of streak plating is required, not just a statement saying streak plating. So if we look at the mark points, we can see mark point 1 requires the use of an inoculating loop to transfer the bacteria. Again, not just saying I will use a loop, they need to have the idea of transferring bacteria with it. The second mark point is for a description or a diagram of streak plating, rather than just stating the term I will use streak plating. Mark point 3 is for a suitable period of incubation. Many candidates stated they would incubate the plates but did not give a time of 24 to 48 hours. Mark point 4 requires a suitable temperature. The temperature range was fairly generous a range somewhere below 30 or below 37. Anything between 20 and 37 was required. Many candidates simply said they would incubate the plates in an incubator but didn't give a temperature. The next mark point is an idea of how you identify the bacteria, the idea of how, by looking at the colour of the colonies or the shape or using selective media. And then the final mark point was often forgotten by candidates, the idea of then using a loop again to transfer colonies onto a separate agar plate. This was the idea of how they would isolate the bacteria. This is an example of an excellent answer, and it gained all 4 marks. The candidate clearly states that they will use a wire loop at the start. Streak plating is described properly. It's not just a named streak plating. Selective media is suggested for how to isolate bacteria, and a correct time and temperature, 30 degrees C here, 24 hours C here, are both given. We'll now go on to looking at question 4b.E2. Now this question was found to be challenging by many candidates. The question requires an explanation of 3 precautions, not just simply stating precautions. Many candidates simply described precautions, which would often be correct precautions, but did not explain the reasons for those precautions. If we look at the mark scheme, we notice that all of these precautions are listed with an explanation. So if we look at the first one, mark point 1, it states that the use of aseptic techniques to prevent contamination or growth of pathogens. Note aseptic technique and its explanation. The second one is again an example of an aseptic technique, such as working near a Bunsen flame, and again we want the explanation to create an updraft or a convection current to carry bacteria away from the plate. Mark point 3 looks for a precautionary temperature, so a temperature under 30, and then a reason because this will discourage pathogens from growing. And then finally, the use of tape that's in a cross rather than sealing the whole plate to maintain aerobic conditions to prevent pathogens. The key with these mark points is all require a precaution and an explanation. This is a very good answer here, and this one gains 3 marks and is a really good example of a precaution listed along with its explanation. If we notice at the start, the candidate states that disinfectants are used on work surfaces. There's the precaution, and the reason then is to kill microorganisms or prevent contamination. The candidate states that we would incubate the plates under 30 as pathogens don't grow at colder temperatures as well. And they've also gone on to state about opening the agar at a slant angle so that the person doesn't contaminate the plate. So in all cases, it's a very good example of a precaution and an explanation. We'll now look at question 6b.E3. This question is, candidates have to evaluate whether the method used in an investigation will provide valid results for water potential of potato cells. This is another practical question, and it assesses candidates' understanding of the strengths of an experimental method. Note the question asks the candidates to evaluate the method. Evaluate requires an explanation of factors that both support and do not support an investigation. There were many excellent answers and a wide range of mark points were given, and we'll look at those on the next slide. The commonest correct answers were cork borers producing equal size pieces, the idea that there is a lack of temperature control, and that discs were often not blotted dry. Here we see the mark scheme for this question. Notice the way it's arranged. There are strengths and weaknesses. If we look at the strengths, we can see that we awarded marks for the idea that the cork borer gave equal size pieces, that discs were cut so that their surface area was increased, The length of time was enough for osmosis. All discs were the same tuber. Many candidates didn't note this and thought that different tubers were used. There was a range of concentrations and it was appropriate to use. Weaknesses were the idea that some of the discs weren't submerged. Discs need to be the same width or size. Repeats need to be done. Only one disc was used in each solution. The discs need to be blotted dry. This was a common weakness that was recognised. More molarities near the intercept point needed to be taken. That the temperature should be controlled. This was another common weakness that was recognised. And that they would need to use a conversion table to actually get osmotic potential. Again, note that we need to get full marks, at least one strength and at least one weakness. Here we've got an example of an excellent answer. The candidate wrote a lot of key points and gained all 5 mark points. If we go back to the mark scheme, you can see that this one has got mark points 3, 1, which are clear strengths, 7, 8, 9, and 10, which are clear weaknesses. The candidate has clearly recognised the word evaluate requires both strengths and weaknesses. This is another example of an experimental evaluation question that asked for candidates to look at a method and then evaluate the method. This one, though, is question 11A.E2. The question focuses on topic 10, which is ecology. And again, like the previous question, requires evaluation. In this case, however, candidates are evaluating the method of collection of data rather than looking at the actual method itself. So this particularly looks at the method of data collection. If we look at the mark scheme again, we see the same layout, strengths and weaknesses. And to gain full marks, at least one strength and one weakness needs to be mentioned. If we look at the strengths, what we see is the experiment showed that there was a lot of data collected. There was a large sample size. It's a simple method, so people would follow it easily. Random sampling was used, so there's a lack of bias. Note that the idea of lack of bias needs to be included here. Weaknesses included that the survey may not have areas that are 1 metre squared. The area may not have actually been chosen at random, or there may have been some bias. This doesn't conflict with mark point 3 because both required an explanation. This mark point here, mark point 6, recognise that some people who are not experts might not be able to identify flowers and plants accurately, and they may not count them properly. The question did not actually state which area was surveyed in the year, and there was no reference to when the time of year was actually taken. So again, we're looking at a method and are being asked to find strengths and weaknesses. Question 7 we'll look at now, and we're going to look at all the parts of question 7. Now this question is a question that was focused on ecological methods from topic 10 and statistics. Candidates were given an example of a plant called hedge woundwort and told that it grows on the edges of woodlands and could be up to 1 metre in height. They're told a student investigated whether light intensity had an effect on the leaf area of hedge woundwort. Now notice this was about leaf area. Some candidates thought it was about populations. They're told that the student measured the area of 20 leaves from plants growing in full sun and 20 growing in shady conditions. And they were asked to then give a suitable null hypothesis. Now, at A level, there are 3 statistical tests that are assessed. These are correlation coefficient, t-test for testing means, and chi-squared for testing populations. Part A.E1 of this question asked candidates to give a suitable null hypothesis. About 1/3 of candidates were able to do this and gave a correct null hypothesis. Where candidates did not, however, give a correct null hypothesis, common errors included referring to correlations. Again, candidates should be clear which statistical test is used in which circumstance. Sometimes they gave a hypothesis rather than null hypothesis. And sometimes candidates referred to light intensity rather than sun and shade. Candidates should also be careful to note that these are often looking for differences, or statistical tests are looking for differences or looking for correlations and not causal factors. Candidates should also always refer to the name of a plant species in a null hypothesis rather than just referring to the size of any leaves. It was common for candidates to state there will be no difference in leaf size without giving any reference to the plant species or the idea of the shade and the sun. In part A.2 of the question, candidates were asked to devise an experiment to compare the leaf areas of plants in sunlight and the shade. We can look at the question and we can clearly see it states, devise a valid fieldwork method the student could use to compare the leaf areas of the plants growing in full sunlight with the leaf areas of the plants growing in shady areas. When devising an experiment, candidates should give details about the method and refer how to make the experiment valid. So they should always think about things like repeats and the controlling of variables. Common errors candidates made were often many used a transect rather than randomly selecting plants in two areas. Many did not refer to hedge woundwort. They often just referred to plants. Candidates should always refer to the plant species they're measuring. Sometimes they did not actually give a method for measuring leaf area. Again, candidates are asked for a method rather than just saying, I will measure the area. Candidates should always make sure that they give full details when describing methods. If we look at the mark scheme, we can see that this is represented there. Mark point 1 is about looking for random selection of plants. Mark point 2 is about measuring leaf area of the named species, hedge woundwort. Mark point 3 is about describing the method of measurement, not just stating that they'll look for leaf area, but saying how they will use something like squared paper or another relevant method. Mark point 4 is about which leaves to select in light and shade, and mark point 5 for controlling a named factor. Here we have an example of a good answer that gained 3 marks. The candidate has clearly stated that they're measuring in a shady area and a sunny area. They are clearly getting mark point 1 for stating the use of random coordinates and quadrats. And they give a method for how to actually calculate leaf area in there. We'll now look at 7b.1, all part of question 7. For this part of the question, candidates need to calculate the size range that would include 95% of leaves using standard deviations. Candidates actually had to select the standard deviations from the text, multiply it by 2, and determine the values of leaf area that were plus or minus 2 standard deviations from the mean. The most common error was finding a value that was plus or minus 1 standard deviation. Candidates should know how to calculate and use standard deviations. Part 7b.2 took the candidates further through the question by asking them to calculate a t-test for the data. The t-test is one of 3 tests listed in the specification, as we've already mentioned, the other 2 being correlation coefficient and chi-squared. This question was generally completed well, and it's clear that centres are preparing candidates with statistical tests very well. The calculated value of t was then used in the final part of the question in 7b.3. In 7b.3, candidates had to use this calculated value to comment on the results of the investigation. Notice the command word in the question, comment on the results of the investigation, and use the information in the table to support your answer. Around a third of candidates gained at least 2 marks, mainly for identifying the critical value correctly and stating that the null hypothesis was rejected and there is a significant difference. Common errors include using the wrong critical value and not referring to a significant difference in leaf area. Many candidates simply stated there is a significant difference, but did not state that it was a significant difference in leaf area. This is an example of an excellent answer that gained all 3 marks. The candidate clearly states that the null hypothesis is rejected. The candidate also explains this by stating that the t-value is greater than the critical value and identifies the critical value of 2.02. They go on to say that there is a significant difference between leaf area in the areas of sun and area of shade. Again, this is a very good detailed answer. They leave no vagueness in there. Weaker answers tended to state there might be a significant difference and did not state that it is between the leaf area in the two areas. When commenting in general on the results of statistical tests, candidates should always make sure that to state that whether they accept or reject the null hypothesis and are clear if the result is significant, giving the full details. For example, there is a significant difference between the mean leaf size in the sun compared with the shade, rather than simply stating there is a significant difference alone. They should always explain that their calculated value is either greater than or less than the critical value and clearly state the critical value. This answer gained 2 marks. The candidate has correctly stated that the null hypothesis is rejected and that there is a significant difference. The critical value, however, has not been identified, so mark point 1 is not awarded. We'll now look at a very few final points across all 3 papers. Generally, the examiners were very impressed with the high standard of scripts. Candidate centres should be really commended for their hard work in preparing these candidates for these examinations. In future series, candidates should think about these points. They should make sure that they read all parts of questions carefully. They should try to maintain a steady pace through the exam and do not feel that they need to fill every line. They should be guided by the mark allocation of questions. They should explore data and information thoroughly when asked to discuss and evaluate, and not focus their answer on 1 or 2 aspects. They should be clear on the meanings of different command words. These can all be found in the specification. They should be fully familiar with the different maths and practical skills listed in the specification. And they should make sure that they're familiar with all the core practicals. They should also, on paper 3, be careful to fully justify or explain experimental modifications if asked. The lack of justifications was one of the commonest errors on paper 3. That brings us to the end of the review of this series of papers. For more courses, you can go to the Pearson Professional Development Academy, and there's a link there on this page. Thank you very much for attending this event. We hope that it's been useful for you and hope it will help you prepare your candidates for future series of exams.